I'm going to talk about protein DNA co-condensation, which something happens due to protein DNA and protein-protein interaction. I'm going to explain that as a mechanism, probable mechanism of genome organization. And I'm working with Sandeep Chaube and Satyavani Bemparala at IMSC. So some of these things Rahul has already introduced you, but I'm going to repeat a little bit. Genome in eukaryotes is organized across scale. So if you look at the lowest possible scale, we have this double helical arrangement of DNA. But if you go all the way to here, at the scale of nucleus, you see these distinct territories occupied by different chromosomes. So if you start from the lowest possible scale, at some level you are going to see these 30 nanometer structures known as chromatin fibers. But most of the time in this talk, I'm going to focus about these regions, which are from few kilo base pair to mega base pair. So here, two possible organizations are proposed, and it's often very difficult to disentangle them. One are these topological associated domains, which are proposed to be formed by loop exclusion models, and another ones are these compartments where you have heterochromatin and euchromatin occupying different compartments. But if you look at across species, what happens, you see some signature differences. So the metric I'm going to talk about genome organization is through these contact maps, which one gets from Hi-C maps. So the high intensity regions are the regions where different regions on the chromatin talk to each other more frequently. So you see those diagonal terms with higher intensity, of course, because one is connected to one. And you see those shadow regions at the background, those are the signature of AB compartments. So you see these regions talk to each other more frequently, not to these. And the residues which are within these regions, they talk to each other more frequently. But if you look at bacteria, you don't see those off-diagonal terms. It's more like homogeneous. And this other diagonal term that goes here is because of the origin of replication is connected to terminus. And then if you look at archaea, you don't see those topological associated domains which are present in eukaryotes. You see more like these big squares. So there are distinct compartments. We don't see loop-like structures here. So this was from a review paper published in 2020. And then the same group did some experiment in archaea. The species is called Solpholobus icelandus. And you see like there are certain diagonal terms here. But also there are some off-diagonal terms here. And this Hi-C-like contact maps can be converted to a map of correlation coefficient, which enhances the contrasts. So you see certain compartments here, and which are like blue and red region, and there are some diagonal terms here. But if they delete one protein, you see there is a drastic reduction in the off-diagonal terms. So the question we started asking, like, how does the presence of one protein increases the off-diagonal contacts? So I'm going to explore this question by some simple physics-based models. So the way we are going to address this, we thought like we look at a simple bead on a string polymer, and we look at three possible variations of this polymer. One where there is only one type of bead, we call that A. In another, there are two kinds of bead, but they are arranged in two block sequences. In the other one, where they are arranged in alternative sequences. So the purple ones are the weak binding sites, and the blue ones are the strong ones. So you have arrangement of half weak, half strong, alternative weak and strong, and a fully weak side. So the proteins are denoted by this yellow molecule C, and they can bind to this A region with 1 kBt affinity, 1.5 kBt affinity, and the B region with 2 kBt affinity. So these are these attractive interactions in my system. And the polymer doesn't like, monomers in the polymer, they don't like each other. So they have a repulsive interaction. So if I don't have any proteins, it will like to just float around. It won't come to contact. And the other thing I'm introducing is this interaction between these C-type molecules, which are like very crude representation of protein in my case. and I increase the concentration of it in two limits, where this protein-protein interaction is weaker than the DNA-protein interaction and where it is stronger than the DNA-protein interaction. So in the first case, what happens, like I have this homopolymeric system, completely A, all the A's, and then I increase the concentration of the protein yellow molecules. And I see the contacts between different beads on the model chromosome. So what happens, you see progressively with increase in concentration, you see higher and higher contacts, which is not very surprising. Like you increase the molecule that brings different segments of the polymer together and you see more and more contact. But if I go to the high protein-protein interaction limit and increase the concentration, something different happens here. The contact intensity sort of go down. So if you look at the leftmost and rightmost picture, the contacts are extremely low here. And the reason we think what is happening here is like if you increase the protein-protein interaction too much, they like each other a lot and not the DNA. So these are some of the configuration you can get from these simulations. So this direction I'm increasing protein-protein interaction, this direction I'm increasing concentration. So low protein-protein interaction, low concentration, the chromosome is open. If you increase concentration, it bridges different monomers together. High protein-protein interaction, low concentration, it's collapsed, but the proteins are sort of everywhere. But if you increase the concentration, they are localized within this DNA. So they form some kind of a droplet-like structure here. So that effectively reduces the contacts between the monomers. The other variation we tried is like take a blocky sequence. And this is the experimental data. And if I take a blocky sequence and look at a particular enzyme in my simulation where the protein-protein affinity is high, but concentration is low, so I'm not in that droplet regime. You see this compartment formation, like this region has more high intensity contacts than this region. And if I zoom into further, the contacts are actually on the off-diagonal term also. They are not only on the diagonal terms. But if I go to the third variation of my sequence, in the same, keeping all other parameters same, we see that difference between these different compartments are negligible. So here in homopolymer, you see these two compartments are of similar intensity. If you have a blocky nature, then you see higher intensity here. And this difference sort of, again, goes away if I make it alternative. So that's sort of the summary of what I have been doing so far, that if you have some heterogeneous DNA sequence and have protein-protein interaction, you can get various structure of chromatin. And if you take a blocky sequence, that gives compartment which are somewhat similar to you see in high synapse in archaea.
Nice talk and nice results also. Can you help me understand also the first problem that you posed, that how does one mutation completely change the landscape? So what was your conclusion in the light of these simulations?
I think these proteins bind to repeated motifs on the DNA, and they also interact amongst each other.
Hello. I wanted to ask, by blocks, do you mean repetitive sequence, like
Right.
Repeats? So you say that if we have a repeat sequence with low protein and if the apologies. So when there is DNA heterogeneity, why? Can you go to the last slide?
Sure.
This one.
This one?
Previous one. Previous one.
Which one?
Where we had one repetitive sequence, two blocks, and then the heterogeneity one.
Is this the one or this one?
Yeah, this one.
Okay.
So, could you please explain why we observe that in alternative?
Okay, so here is what happens, like all of them are one kind. And it's like this matrix is sort of symmetric. So all the information you will have is, you can think of like the lower triangle part is the same as the upper triangle part. So all information are here only. So if you look at this here, this thing, this compartment, whatever contacts are there, they are not very similar to here. But if you have a blocky thing, so my sequence 0 to 1000 is this pink one. But from 1000 to 2000, you have this blue one. And this blue one binds to the yellow ones with sort of higher affinity.
So here, the pink one is one type of nucleotide and the blue one is the other type of nucleotide.
Right, that's the assumption.
But the protein is the same.
The protein is the same.
I see. Okay. Thank you. Sorry.
So we know that the TAD boundary, the formation of TAD facilitated the interaction between different chromatin regions.
Sure.
So does TAD also facilitate the interaction between proteins and the DNA, the special motifs on the DNA?
I mean, they bring a distance element close to each other by this loop extrusion process. But I'm actually don't know. I'm not sure about if they can bring protein DNA together. But it can happen like you have proteins of different types, and interaction among them can bring DNA protein together.
Yeah. So you have homogeneous block and alternative. Do you have like a parameter which will tell you that this particular interaction matrix is homogeneous or this is block? I mean, how do you know that this is homogeneous in block and alternative? Because
how we know that this one is closer to experiment and not this one? Is this the question?
How do you differentiate between them?
Oh,
like, is there a
right. So I'm not shown you the quantitative metric that one can extract from these maps. So what you can extract is, as a function of this residue length, the length along the contour, what is the probability of contact, and the exponent sort of shows you the quantitative difference. So the exponent here will show you like this as a different, you can fit it with like a 2 exponent thing. Here it falls more easily, here it remains like shallow. So you get different exponent in this contact probability. So experimentally something like 0.4 is observed, and I sort of compare that with my model.